Memory is one of the most fascinating and essential functions of the human brain. It occurs over different scales of time and involves intricate networks of neurons across various brain regions. Memory is integral to who we are, as we are made from many experiences we encounter in life. The importance of memory is so clear when we are confronted with the disastrous implication of a loss of personality in patients suffering from memory loss. But how exactly are memories formed and stored over long periods? How do groups of neurons store a memory trace in the network? How is the memory secured and how is it reactivated? In Berlin, the Deutsche Forschungsgemeinschaft is funding the Collaborative Research Center 1315 on mechanisms and disturbances of memory systems. In this consortium, many laboratories are investigating these questions. The role of our team is to design and manufacture cutting-edge tools that are then used by other teams of the consortium to explore and manipulate memory circuits. Welcome to our laboratories at the Charité Campus Mitte in Berlin. Here we are passionate about understanding the complex circuits in the brain that make memory possible. But to truly grasp how memories are formed and stored, we need to peek inside these circuits and even interact with them.
That's where our work comes in. We are a team of 3 scientists, Christian Wosenmund, Johannes Fjöberg and myself, Benjamin Rost, who develop and customize optogenetic tools, viral vectors and gene transcription readouts. And thereby we provide highly specialized molecular tools that neuroscientists can use to investigate the formation and retrieval of memories.
Optogenetics is at the heart of our research. This is a technique that uses light-sensitive proteins from nature, so-called photoreceptors, to control neurons by light. In the most straightforward case, light-guided ion channels are used to switch neurons on and off, what is possible with high temporal and spatial precision. In our lab, we explore and engineer these photoreceptors to tailor their biophysical properties for their final application in neurons, and for example tune the activation kinetics and color. By shining different colors of light, we can then control multiple pathways simultaneously and explore not just where neurons are located, but also where they're projecting to and how they're contributing to memory formation.
Imagine trying to understand how a complex machine like a car works. You would need to look inside, see how each part functions, and observe how they all work together. You may want to inactivate one part to see what happens then, or you may want to activate another part at a certain time point to investigate the reactions. We can now do the same with the brain's memory circuits using our molecular tools.
By selectively inhibiting or exciting neurons, it becomes possible to test how changes in neuronal activity affect memory formation and consolidation. One way to do this is to perform optogenetic experiments which involve expression of light-activated proteins in neurons. This we can do by using modified viruses that encode a light-activated protein. We can design these viruses to express the genes only in specific type of neurons or in specific areas of the brain. And these viruses also encode fluorescent marker proteins, which allows us to visualize these neurons under the microscope. Over the years, we learned a lot about how we can tailor our viral messengers and the optogenetic tools to the specific needs of each researcher. In the past, optogenetic tools acted as simple on or off switch for neuronal activity. But with our latest approaches, we can now specifically modulate individual molecular signaling cascades that play a role in memory formation. And to become even more precise, we restrict this modulation to individual parts of the neurons, for example, the axon or the dendrites.
As you already learned, viral tools are very effective for memory research and indeed for many other areas of basic and clinical research. To ensure the highest safety standards in viral tool production, we established a dedicated laboratory 10 years ago. This facility was designed specifically to develop safe viral vectors, serving not only the Charité campus, but also the wider life science community in Berlin and throughout Europe. Since worldwide research continues to improve and broadens the use of viral tools for research, we continuously adopt our tools to implement new cutting-edge technologies. For instance, In response to requests from our users, we expanded recently to novel viruses that now enable us to map the connections from one neuron to all the neurons it sends information to, thus enabling us to even better map the traces of memory.
Sometimes the details matter. For example, we might need to inhibit a neuron quickly and briefly, or over a longer period. We develop different optogenetic tools modulating inhibitory membrane fluxes for potassium and chloride, or activating entire G-protein-coupled signaling cascades.
This research not only will help us to understand how we form and maintain memory throughout our life, this knowledge will also help us in the long term to better understand and treat diseases related to memory loss, such as Alzheimer's disease.
We also believe that it is important to collaborate with all other researchers worldwide, and we actively support exchange of our tools we designed by sharing them through our virus production facility. We think this will help a wide community of memory research in utilizing modern molecular technologies.
The journey to understand memory is complex, but with innovative tools and collaborative efforts, we're making strides towards unlocking the secrets of the brain. At the SFB1315 project, we are not just studying memory, we are providing the keys for others to explore and discover.